Hi, hello, good morning. This is Ramesh Kumar Lalkota, Assistant Professor of Botany, MBS Government Arts and Science College, Autonomous, Nagar, Telangana State. In our last class, we studied about electron transport system. We are studying respiration in plants. We saw glycolysis. Glycolysis takes place in cytoplasm, and the end products of glycolysis are pyruvic acid, two molecules of pyruvic acid, and net gain of 2 ATP and 2 NADPH are formed in glycolysis. Glycolysis takes place in cytoplasm. Then the pyruvic acid, which is a 3-carbon compound, enters into mitochondrial matrix. There it undergoes oxidative decarboxylation to form acetyl coenzyme A. Okay, here also each pyruvic acid on decarboxylation and oxidation, it produces one NADPH is reduced here. Okay, so for two pyruvic acid molecules, two NADPH are formed in oxidative phosphorylation of oxidative decarboxylation of pyruvic acid. Okay, so this acetyl coenzyme A is the connecting link between Krebs cycle and glycolysis. Acetyl coenzyme A enters into Krebs cycle where it undergoes four oxidation reactions and two decarboxylation reactions, as a result of which three NADPH, one FADH, and one ATP are formed. Okay, so we have two acetyl coenzyme A molecules. So as a whole, six NADPH, two FADH, and two ATP are formed in Krebs cycle. So these are reduced compounds. NADH, FADH are reduced compounds. They are rich in energy. Okay, so these NADH and FADH undergo oxidation, and they give the electrons to the oxygen. The ultimate receiver of electrons is uh, oxygen, which undergoes reduction into water, okay, which reduces to water by accepting electrons from these um, reduced NADH and FADH and protons from mitochondrial matrix. The oxygen undergoes reduction into water molecule. That's what happens during electron transport system, and during these processes a proton gradient exists, and this proton gradient um, acts as a proton motive force, and which ultimately results in the formation of ATP. And this process is explained by chemiosmotic uh, theory proposed by Mitchell Stark. Okay. Um, then Peter Mitchell. Peter Mitchell was the person who explained this electron transport system. Okay. So now uh, we will have a glance. Electron transport system, we need to understand these components before entering into electron transport system. Multiple enzyme complexes, we said that we have 1 to 5 multiple enzyme complexes. Okay, the first 1 to 4, they help in electron transfer, and the fifth one for ATP production. Okay, then mobile electron carriers, we have ubiquinone and cytochromes, proton translocation sites, number of protons translocated, and mode of ATP formation. We saw that complex 1 is NADH dehydrogenase or ubiquinone oxidoreductase, and it has FMN as cofactor, and complex 2 succinate dehydrogenase or succinate Q reductase, it has FAD as cofactor, and it transfers electrons from succinate to ubiquinone. Complex 3 cytochrome C reductase or ubiquinol cytochrome oxidoreductase, and it contains a cytochrome B and cytochrome C, which are heme proteins, and they transfer the electrons from iron sulfur protein to cytochrome C via cytochrome C1. And the fourth one, cytochrome C oxidase, here cytochrome A and cytochrome A3 are the electron carriers, and the electrons are transferred from cytochrome C to cytochrome A3 via cytochrome A, which are ultimately transferred to oxygen from cytochrome A3. These are the multiple enzyme complexes which take part in uh, electron transport. We said that, you can see here, this is complex 1, this is complex 2, this is complex 3, and this is complex 4. All these are present in inner membrane of mitochondria. This is the inner membrane of mitochondria. Okay, so this is the inner membrane of mitochondria, and this is the matrix towards outside, intermembranous space, and uh, outside to the intermembranous space is present outer membrane. Okay, so these are the proteins. Enzymes are nothing but proteins. They found embedded in the inner membrane of mitochondria. So NADH and FADH are formed as a result of glycolysis, Krebs cycle, and oxidative decarboxylation of pyruvic acid. So these NADPH and FADH here, they undergo oxidation, releasing protons and electrons. Okay, so in the first step, this is the first enzyme complex. Here NADH undergoes oxidation. See, H plus ions and electrons are released from this, and it converts into NAD, and this takes place in the presence of NADH dehydrogenase or ubiquinone oxidoreductase enzyme. Here FMN is the coenzyme which accepts the electrons initially, and through this FMN the electrons reach ubiquinone cycle. Okay, this is ubiquinone cycle, and from FMN to ubiquinone, iron sulfur proteins carry the electrons. For every 2 electrons, 4 protons from matrix enter into intermembranal space. This is the intermembranal space. So for every 2 electrons, 4 protons from matrix enter into the intermembranal space. Now, by accepting the electrons, ubiquinone undergoes reduction to ubiquinol. Ubiquinone to ubiquinol. Okay, so it undergoes oxidation again. Initially, it accepts the electrons, undergoes reduction to ubiquinol. Now, it releases the electrons, undergoes oxidation. Okay, so these electrons are transferred to cytochrome B, that is complex 3. Okay, cytochrome B, they reach the cytochrome B. From cytochrome B to cytochrome C1. Okay, so during this process also, 4 protons enter into intermembranal space. Okay, during the transfer of electrons from cytochrome B6 to cytochrome C1, 4 protons from mitochondrial matrix enter into intermembranal space. So here 4 protons and here 4 protons. Okay, then from cytochrome C1, these electrons transfer to cytochrome C. Here also it undergoes oxidation and reduction reactions. First it was in oxidation state. It takes 2 electrons, undergoes the reduction, and it again releases the 2 electrons by oxidation, and these electrons are received by cytochrome A. Okay, these electrons from cytochrome C are transferred to cytochrome A of complex 4, from cytochrome A to cytochrome A3, and ultimately to oxygen. Okay, here also 4 protons out of 4, 2 protons reach intermembranal space, and 2 protons are utilized for reduction of this oxygen. Oxygen receives electrons from cytochrome A3, that is complex 4, and 2 protons from mitochondrial matrix, and it reduces to water. So ultimately these electrons are transferred via various enzyme complexes to oxygen. Ultimately it is reduced to water. And during this whole processes, see here 4 here, 4 protons here, and 2 protons there. A total of 10 protons enter into intermembranal space. And we have enzyme complex 2 also. Okay, so the FAD formed in Krebs cycle, it doesn't enter into complex 1, it enters into complex 2. Here you can see that from complex 2, where FAD is primary acceptor of these electrons. Okay, this is a coenzyme, and this FAD, from this FAD, electrons reach ubiquinol, and from here on the process is same, but except that these 4 protons from the first step won't enter into the intermembranal space, and from here on 4 plus 2 protons only enter into the intermembranal space, that is only 6 protons when FAD enters through complex 2 only 6 protons enter into the intermembranal space. In the same way, see, this is in uh, inner membrane of the mitochondria. Okay, so cytosol NADH are also there, which are formed during glycolysis. Glycolysis takes place in cytoplasm. So these NADH, they won't enter into the mitochondrial matrix. It won't allow to enter into the mitochondrial matrix. So what they do is they transfer the electrons to FAD. They undergo oxidation by releasing electrons, and FAD receives the electrons and it undergoes reduction. So this FAD enters into complex 2. That's why the cytosol NAD NADH produces only 6 protons. It transfers, while the electrons from these NADH reach the oxygen, only 6 protons enter into intermembranal space. Okay, so for every 3 protons, 1 ATP formation takes place. So for NADH, we have a total of 10 protons. So out of 10 protons, 9 protons are used for production of 3 ATP, and for cytosol NADH and FADH, only 6 protons enter into intermembranal space. So only 2 ATP are formed for cytosol NADH and FADH. Okay, so how these ATP formation takes place, we will see. Okay, so here, due to accumulation of H plus ions, there is a high concentration of H plus ion in the intermembranal space, which creates a proton gradient. Here pH decreases and the concentration of H plus ions increases, pH decreases, and a proton gradient exists between intermembranal space and matrices. So this higher concentration of these protons creates a proton gradient, which acts as a proton motive force. So these protons, they won't this membrane won't allow these protons to pass through them. So what happens here, the role of a fifth enzyme complex, which is known as ATP synthase, comes into action. It is present in the mitochondrial inner membrane. It is embedded. Okay, so it has two parts, the F1 region and the F0 region. F0 region is a stalk, and it is present embedded in the inner membrane, and F1 is a head, which is present towards the mitochondrial matrix. Okay, so this acts as a channel for flow of these protons. So here the concentration of protons is very high. So due to repulsive forces or proton gradient or proton motive force, these protons flow from this channel and they're liberated into the matrix. While they move from this channel, they create some current. So this current helps the head of this F0 F1 particle to rotate, and while rotating, it generates some energy, and this energy is utilized by ADP and inorganic phosphate present in the matrix, and ADP is converted to ATP. This is oxidative phosphorylation. That is, uh, we saw that photophosphorylation in light reaction, and substrate level phosphorylation in glycolysis and Krebs cycle. Now here it is oxidative phosphorylation. Because of oxidation reactions, this flow of protons is possible. Okay, during the transfer of electrons only, these protons enter into intermembranal space. Okay, so during the oxidation, this happens. That's why this is known as oxidative phosphorylation. So ATP are formed for every 3 protons, 1 ATP formation takes place. So this is known as chemi-osmotic hypothesis proposed by Peter Mitchell. Okay, then we saw it yesterday. Okay, I think you might have understood the concept of electron transport system. So as a whole, we saw that the balance sheet, see, glycolysis 4 ATP NADPH, out of which 4 ATP, 2 ATP are utilized, 6 is the net gain. And during oxidative decarboxylation of pyruvic acid, we have uh, 2 NADH formed, so 2 into 3 equal to 6, and in Krebs cycle we have 2 ATP and 6 NADH and 2 FADH, a total of 24, and the net gain is 36. Okay, so each ATP carries 7.6 kilocalories of energy. So this 7.6 calories of energy into 36 ATP gives 273 kilocalories of energy. Okay, so 276 kilocalories of energy is formed by this ATP. And then we said that on complete oxidation of glycolysis, we have 686 kilocalories of energy. But here, when we count these 36 ATP, each ATP 7.6 kilocalories. So we are getting only 273.6 kilocalories of energy. So where is the remaining energy go on? So this remaining energy is wasted in the form of heat energy. Okay, so now if one molecule of glucose is burnt in a calorimeter, about 686 kilocalories of energy is liberated. Okay, then we have only 36 ATP, and this 36 ATP into 7.6 gives 273.6 kilocalories. Therefore, if we find out the efficiency of the biological oxidation, then we will get near about 40%. Only 40% of the energy is stored in the form of ATP, and the remaining 60% is wasted in the form of heat. Okay, now we will see the next topic, that is mechanism of anaerobic respiration. What is anaerobic respiration? If respiration takes place in absence of oxygen, then we call it as anaerobic respiration. Okay, in higher plants, glucose molecules are partially oxidized, resulting in the formation of ethyl alcohol and carbon dioxide. Okay, the glucose is first converted into pyruvic acid by EMP pathway or glycolysis, where see here, you can observe here, glucose on glycolysis forms two pyruvic acid molecules. Here, two ATP formation takes place, two net gain, and two NADH formation. We already saw that in glycolysis, two ATP and two NADPH are formed. Okay, so the ultimate products formed during glycolysis are two molecules of pyruvic acid. This pyruvic acid is broken into ethyl alcohol and carbon dioxide in anaerobic respiration. This takes place in two steps. First step, decarboxylation takes place, and in second step, uh, again dehydrogenation or reduction reaction. Okay, so in the first step, decarboxylation of pyruvic acid, as a result acetaldehyde formation takes place. Okay, so here two molecules of pyruvic acid liberates two molecules of carbon dioxide, and two molecules of acetaldehyde is formed, and this reaction takes place in the presence of the enzyme called pyruvic acid decarboxylase. Hmm? This is the first step, and in the second step, the acetaldehyde molecules are then reduced to ethyl alcohol. See, this acetaldehyde reduced, and the reducing power here is NADH, which is formed during glycolysis. Okay, this NADH is utilized here, and this reduction, the aldehydes on reduction gives alcohols. So two acetaldehyde molecules ends up in two molecules of ethanol on reduction, and the reducing power utilized is NADH. It undergoes oxidation to NAD, and the protons are supplied by this NADH to this acetaldehyde, and the end product is ethanol. So as a result of anaerobic respiration, we have two molecules of carbon dioxide and two molecules of ethyl alcohol. Glucose on a partial oxidation ends up in carbon dioxide and ethyl alcohol. This is anaerobic respiration. Now we will see respiratory substrates. What are respiratory substrates? The organic substances which are oxidized in cellular respiration for releasing energy are called respiratory substrates. And examples for these respiratory substrates are carbohydrates, fats, and proteins. Okay. Uh, Now, the respiratory substrate, among the various substrates available in cells, the most prominent ones are carbohydrates and fats and organic acids. Okay. Uh, Under natural conditions, only carbohydrates like sugars and starch are used up in respiration. That is why in many plant tissues, the respiratory quotient is maintained as 1. And this type of respiration is called floating respiration. Normal conditions, the respiration is called floating respiration. If the carbohydrates are completely used up, other protoplasmic substrates like proteins, amino acids, or fatty acids are used as respiratory substrates. Then it is known as protoplasmic respiration. Okay, so if the carbohydrates are used as respiratory substrates, what happens we will see. Okay, so um, these are the principal respiratory substrates in the cells of higher plants. Okay. The most important among them are glucose, fructose, starch, glycogen, and sucrose. Glucose acts as a respiratory substrate directly. Fructose also directly acts as a respiratory substrate, but starch, it is hydrolyzed to glucose 6-phosphate first. Then sucrose, it is hydrolyzed to glucose and fructose before undergoing this, before acting as a respiratory substrate. First, they convert into glucose or fructose. These glucose and fructose alone, they directly acts as respiratory substrate, but the starch, glycogen, and sucrose, they first convert themselves to glucose or fructose, then they acts as respiratory substance. Then fats. Sorry. These fats, they undergo hydrolysis in the presence of lipase. They are converted into fatty acids and glycerols. Fatty acids, they first form into acetyl coenzyme A, and this acetyl coenzyme A enters directly into the Krebs cycle. In the same way, glycerol is also converted to pyruvic acid, and this pyruvic acid undergoes oxidative decarboxylation, resulting in the formation of acetyl coenzyme A, and this acetyl coenzyme A enters into Krebs cycle. Then proteins. Proteins, they are hydrolyzed into amino acids, and later on by deamination process, these amino acids are converted into various organic acids, and these organic acids enter into the respiratory, that is Krebs cycle. Okay, there are number of organic acids in the Krebs cycle, no. So these organic acids which are similar to those, they enter into the Krebs cycle directly. First, proteins are hydrolyzed to amino acids, then amino acids undergo deamination reaction, then they form into other organic acids. These organic acids enter into the Krebs cycle. Okay. Then respiratory quotient. What is respiratory quotient? The ratio between carbon dioxide liberated and oxygen consumed by a given weight of tissue in a given period of time at NTP is known as respiratory quotient. That is RQ equal to volume of CO2, carbon dioxide, evolved by volume of oxygen absorbed. Hmm? Um, and the value of this respiratory quotient for a tissue depends upon the type of respiratory substrate which is oxidized in respiration. If respiratory substrate carbohydrate, then the RQ value will be different. If it is fats, then it is different. If it is proteins, then it is different. Then if it is organic acids, in such a way, the RQ value of a tissue depends upon the respiratory substrate of that tissue. Okay, if carbohydrates are the respiratory substrates, then we will see what is its RQ, respiratory quotient. Okay, when hexose sugars such as glucose or fructose are oxidized, the volume, see the volume of oxygen absorbed, 6 molecules of oxygen is observed here, and 6 molecules of carbon dioxide is liberated or evolved. So RQ of glucose is equal to 6 molecules of carbon dioxide by 6 molecules of oxygen. So 6 get cancels, and the RQ of glucose is always 1. So all the carbohydrates has the RQ value equal to 1. If fats used as respiratory substrate, fats are poorer in oxygen as they are in a more reduced state than carbohydrate. Therefore, they require more oxygen for complete oxidation, and the RQ value always is less than unity. See, the evolved carbon dioxide molecules are less than the observed oxygen molecules. You can see here for oxidation of this tripalmitin fat, 145 oxygen molecules are required, and evolved carbon dioxides are only 102. So 102 by 145, it gives 0.7. So all the fats in a similar manner, they have the RQ value of 0.7. In the same way as these fats, the proteins also have 0.7 to 0.8 as the RQ value, because they require more oxygen for oxidation. So RQ value of fats and proteins, fats 0.7, proteins 0.8. The RQ value of organic acids. See, here organic acids have uh, more oxygen content than other substrates. So they require very less oxygen for the oxidation, and liberated carbon dioxides are more than observed oxygen molecules. See here, malic acid, we have oxygens more than carbons, more oxygens than carbons. So they require less oxygen for the oxidation, and the liberated carbon dioxides will be more than this oxygen observed. So RQ value of malic acid comes to 1.33. All organic acids greater than 1, more than unity. Okay, and RQ value of succulents. See, here, mm, mm, uh, number of aliphatic carboxylic acids like citric acid, malic acid, succinic acid, intermediate reactants in the sequence of reactions in Krebs cycle. So they are found in considerable amounts inside the cells. In certain plants, as Crassulaceae and Cactaceae, succulent plants, okay, organic acids are synthesized in large quantities, but these are again decomposed during the day. They absorb the carbon dioxide during nights. Stomata are open only during nights because they are xerophytic in nature. To preserve water, their stomata are closed during daytime, and they are open only during night time. So they absorb carbon dioxide during night time, and they fix this carbon dioxide in the form of organic acids, and these organic acids, they liberate this carbon dioxide during daytimes, and this carbon dioxide enter into um, uh, C3 cycle to fix as carbohydrates. Okay? That is, they enter into photosynthesis. Carbon dioxide, which is stored in the form of organic acids, is liberated during daytime, and that enters into photosynthesis. Okay, so the dark acidification and light deacidification is usually referred to as Crassulacean acid metabolism. Okay, uh, so here you can see that during daytime, see, observed oxygens are 3, but no carbon dioxide is released here. Whatever carbon dioxide, it is utilized during the daytime. If the carbon dioxide is liberated, it won't be released outside. Okay, so mm, only oxygen is observed, but no carbon dioxide is liberated. 0 molecules of carbon dioxide by 3 gives 0. Always RQ value of succulents is 0. Organic acids 1.33, proteins 0.8, fats 0.7, and carbohydrates it's 1. This is about uh, uh, RQ values, respiratory quotients. Okay, so one more time, RQ value of succulents. Hmm? So here the members of cactus in Crassulaceae, when placed in darkness, they show incomplete oxidation of carbohydrates. As a result of this, accumulation of organic acids like malic acid, citric acid, and oxalic acid takes place. Okay, during this process, oxygen is observed without the evolution of carbon dioxide. Carbon dioxide is not evolved, only oxygen is observed. So the RQ value is always 0. You need to remember this. In succulents, the RQ value is always 0. Organic acids more than 1, and carbohydrates 1, proteins 0.8, and fats 0.7. You need to remember all these things. Okay. Then fermentation. Fermentation, this is a anaerobic respiration again, but sometimes fermentation also takes place in the presence of oxygen, that is in yeasts and bacteria it takes place in presence of oxygen, but oxygen it won't involve in this process. Okay, but in higher plants it takes place in absence of oxygen. uh, so this is an anaerobic respiration. In higher plants, no oxygen is present. If it is present also, anaerobic respiration takes place in yeast and bacteria. So fermentation, we cannot call it as anaerobic respiration. It takes place in presence of oxygen or in absence of oxygen. In higher plants, it, it takes place only in the absence of oxygen, and in lower organisms such as yeast and bacteria cells, if oxygen is present, also no problem for anaerobic respiration. Okay, so here what happens is of two types: alcohol fermentation and lactic acid fermentation. Alcohol fermentation, what happens? It takes place in two steps, as we saw that. First, decarboxylation and then reduction, resulting in the formation of ethyl alcohol and carbon dioxide. In the same way, lactic acid fermentation, this takes place in muscles, and as a result, lactic acid formation is lactic acid forms as a result of the lactic acid fermentation. Yeah, again, lactic acid fermentation is of two types: homolactic fermentation and heterolactic fermentation. In homolactic fermentation, only lactic acid molecules are formed, but in heterolactic fermentation, a molecule of ethyl alcohol, lactic acid, and carbon dioxide is formed in heterolactic fermentation. So, fermentation here, two types: alcoholic fermentation, which is similar to that of anaerobic respiration, as a result of which ethyl alcohol and carbon dioxide formation takes place. And one more type of fermentation is lactic acid fermentation, and it takes place in muscles. As a result of lactic acid fermentation, lactic acid formation takes place. Okay, so here uh, the predominant end product of is lactic acid in lactic acid fermentation. Glucose yields two molecules of pyruvic acid through the glycolysis process. This pyruvic acid is reduced to lactic acid by the enzyme called lactic acid dehydrogenase. This lactic in lactic acid fermentation, here the pyruvic acid, which is formed as a result of glycolysis, is mm, uh, reduced to lactic acid in the presence of lactic acid dehydrogenase enzyme. Okay, so here NADH is utilized for reduction of this lactic acid, reduction of pyruvic acid to lactic acid. Two molecules of lactic acids are formed from each pyruvic acid, one lactic acid. We have two pyruvic acids, so two lactic acid formation takes place here. Okay, then um, in heterolactic fermentation, what happens? Glucose ends up in pyruvic acid, and this pyruvic acid undergoes reduction, and during this process one molecule of ethyl alcohol and one molecule of lactic acid with the liberation of carbon dioxide. It is heterolactic uh, fermentation. Okay, this is about fermentation. This is all for today. So I hope you understood what I said. Thank you very much for listening to me patiently.